Hi guys, Miss Francis here. Last time I made a video, we were talking about the DNA technology applications to agriculture. So we're going to continue there, where transgenic organisms can be created using various modern technology approaches. A transgenic organism can also be called a genetically modified organism. So we're going to talk about how I can actually clone an organism. One of the methods is called somatic cell nuclear transfer, where we know that a somatic cell is a body cell. So how did scientists make Dolly the sheep? Well, what they did is they took a donor cell from a sheep's udder, and they're going to take this nucleus from the donor cell and insert it into an egg cell. So an egg cell is taken from an adult female sheep, but the nucleus of that egg cell is removed, and now I just have the egg cell with my donor nucleus, and they're going to be fused together using an electric shock. Now that fused cell begins dividing normally into an embryo, where that embryo can then be implanted in my surrogate sheep. The embryo develops normally into, in this case, a cloned lamb, where my cloned lamb is an identical copy to the nucleus that was taken from my donor cell. Now, what's the difference between cloning an organism versus cloning a gene? Well, when I'm cloning an organism, I'm making a copy of the entire organism, but if I'm cloning a gene, I'm just copying one of the organism's genes. Now, cloning a gene can still be used in creating transgenic organisms or genetically modified organisms. Well, how? Because agricultural scientists can engineer crops that have desirable traits, such as the trait for delayed ripening or the trait to resist spoilage and disease. I might have a tomato plant that I want to insert the gene, the antifreeze gene, so that my tomato plant doesn't freeze in cold weather. So foreign genes can be inserted into a plasmid using recombinant DNA techniques, where I produce an organism, in this case a plant, that has a new desirable trait. So in the past few years, roughly half of the soybeans and corn in America have been grown from these genetically modified seeds, where there may be seeds that have genes for resistance to weed-killing herbicides or to infectious microbes or pests. Scientists have also been using gene transfer to improve the nutritional value of crop plants. For example, creating transgenic rice that produces beta-carotene, where beta-carotene is used to make vitamin A. There are ethical implications of creating these transgenic organisms because we don't fully yet know the ramifications that these transgenic organisms can have on humans. So, for example, does eating this transgenic rice have some sort of unintended consequence? And we just don't know yet. So that's our discussion on cloning organisms and transgenic organisms.